Okay, thank you very much for the welcome. It's a great pleasure to be here. So yeah, as I said, so I'm Mark. I'm based in Norwich at the Norwich Research Park, and I lead something called the Centre for Microbial Interactions. And this is a grouping of microbiology research groups across all the different institutions that are based on the research park. So we have well over 100 different microbiology research groups studying a whole bunch of different things. And I'm going to try and use a few examples of things we're doing to hopefully convince you that microbes are interesting and important, and that they do rule the earth, but we'll see. So, just to start with, what does microbe mean? Just very, very simply, what are we talking about? Well, it's from the Greek, and it literally just means small life. Okay, so when we're talking about microbes, we're just talking about things you can't see with your naked eye. Okay, so stuff that you need a microscope to see. So, until the microscope was invented, we were somewhat naive to the fact that actually most of the life on Earth we can't see. In fact, the great majority of life on Earth is microbial, and we'll explain a bit more about that and the things that they do and why they matter. So, what does that mean? Small things? Well, this is a kind of scary-looking diagram, but it isn't. This is a family tree of all the life on Earth. Okay, so all the organisms that we know about. You can take bits of their DNA, and there's some things that are shared by everybody, and you can use that and you can compare the sequences, and then you can make a tree to see who is most related to who. And when you do that, you see that this is a representation of basically all of the life on Earth. And this little green bit here is the eukaryotes, and we are a little tiny bit of the green bit. And down here is where you find the birds, the bees, the bananas, people. We are a little offshoot of this green bit. Everything else is microbial, okay? So microbes really do rule the earth. They are almost all of the life on Earth is microbial one form or another, and including a bunch of this eukaryotic stuff, actually. So we are a tiny fraction of the diversity of everything that we know about, on Earth at least. And probably the rest of the universe, but who knows. Okay, so when we talk about that, what does that mean? What does this great diversity actually break down to? Well, microbes come in different sizes, shapes, types, and there are very different kind of microorganisms. And we're just going to talk a little bit about some of these just to kind of prime some of the things we might talk about later. We've probably heard about bacteria. So bacteria, things like MRSA or E. coli, these are organisms that are free-living. They live on us, they live in us, they live in the environment, they're pretty much everywhere, and they do all sorts of important things. We have the archaea. So the archaea look like bacteria, but they're not. They're a different kingdom of life, and actually we have evolved kind of down a line after them. And they do really interesting things. So they include bugs called the extremophiles. So these are organisms that can survive really extreme environments. So super cold, super hot. So you find these things in kind of Yellowstone Park in the geysers. There are bugs that can grow at the very high temperatures there. There are bugs that can grow at the huge pressures at the bottom of the sea. There are some bugs that can survive the high levels of radiation in space. Okay, so these are the extremophiles. So really interesting. We have fungi. So fungi, these are the molds, the yeasts, the mushrooms, things like that that we've all probably heard about. And we use these for various things. So we use them to make bread. We eat mushrooms. We use them to make beer. So these are useful for all sorts of things. In fact, the largest organism on Earth is probably a fungus. So there's a thing called the humongous fungus, which is which was named in America, may not surprise you. And this is based in Oregon, and it's underneath a forest, and it's covering thousands of acres, and it's one organism. It's tens of thousands of tons in weight. It's thousands and thousands of years old, but it's one individual. So some of these fungi can be enormous, and it's one individual that's spread all the way underneath this forest. We have protists, so these are things like amoebae, often found swimming around in water, eating other things. And we have viruses. People can argue, and it's not an argument to have now, about whether viruses are alive or not. They kind of are not free-living without another cell, but they certainly can replicate. So we'll keep them in what we're going to talk about today. And everyone's heard of viruses. We know all about coronavirus, HIV, viruses that cause problems to people. But actually, the most abundant organism on Earth by far would be something called a bacteriophage. So this is a virus that infects bacteria. There are many, many more bacteria than people, and they have viruses too. And actually, about a third of the bacteria in the sea every day are killed by bacteriophage and then turned over. So there's this enormous diversity of bugs. So these are the kind of things we're talking about when we're talking about microbes. So there's this enormous diversity of different types of organisms. They're really different to each other. They do lots of different things. They're everywhere, and we should care. Why should we care? Well, they affect our lives in positive ways, but also negative ways. And I'm going to talk a little bit about some of that in 3 different lenses. So again, work we do in Norwich, we're really interested in 3 areas. We do lots of things, but we're particularly interested in how microbes contribute to a healthy planet, healthy plants, and healthy people. So we're going to go through that as the structure of the talk, really. How do bugs affect the planet, plants, and people, with a few little examples of that, and then we'll kind of wrap it up. Okay, so healthy planet. This is a very famous picture that NASA took a long time ago, in the 70s, I think, actually, the kind of blue marble looking at planet Earth. Of course, the Earth hasn't looked like that for lots of its time. And the Earth is old. So the Earth is 4.6 billion years old, something like that. And if you look at this kind of timeline of life on Earth, you can see all sorts of things have happened. So kind of plants emerged at some point, animals emerged, dinosaurs turned up. And humans, we've been here for the last few 100,000 years. So in the scheme of things, we've been here for the last couple of seconds, right? If you turn this into a 24-hour clock, we've been here for almost no time whatsoever. Microbes, of course, were here first. So they were here kind of 4 billion years ago or so. The first life on Earth is microbial, and they've been here ever since. Okay? So they've been here throughout, and they are intimately associated with all these other forms of life. So none of these things would happen without microbes being there, and microbes affect these in many, many, many ways. Okay. So they've been here all the time, but they're not doing nothing. So they're having profound impacts on the Earth. So one thing I'm just going to talk about is something called the Great Oxidation Event. So this happened kind of 2.5 billion years ago. Okay. So we are all here breathing oxygen in this room. Otherwise, we would be in trouble. But 2.5 billion years ago, there was very little oxygen in the atmosphere. In fact, pretty much none that was free. Okay. Then something happened. And essentially, a bacteria evolved called a cyanobacteria. And the cyanobacteria are incredibly important to life on Earth. They're still here today because what they evolved the ability to do was take sunlight and take carbon dioxide and water and turn it into sugar that they could eat. So that's what they were doing. That's photosynthesis. And when they do that, they break the water, they release oxygen. Okay, so they're starting to release oxygen. So there's plenty of oxygen on the Earth, but it was all bound up in the core, in minerals in the surface or in the water. They started releasing this oxygen as a gas, and over hundreds of millions of years, it then accumulated in the atmosphere. Okay, so that's why we have oxygen on Earth. Now that was the first mass extinction event on Earth. So at the time, almost all life on Earth was killed. Okay, oxygen is actually very poisonous. It's very reactive. It's a very reactive element. It's very poisonous. If you haven't evolved to deal with it, it's very, very, very dangerous. So actually, suddenly, all this oxygen appeared, and almost all life on Earth that was not used to it, was anaerobic, died. Okay, but there were some positives to this. So when this oxygen was produced, it produced an ozone layer that protects from the radiation from the sun, and it produced all of this oxygen that could then be used in aerobic respiration. That wasn't a thing that existed beforehand, and that's what higher organisms rely on. The energy we need to live and survive needs aerobic respiration. So this is only because these cyanobacteria started producing this oxygen. So they caused a mass extinction event, but they also allowed us to live in the world as we know it today. They also generated a lot of global cooling because actually the oxygen in the atmosphere broke down a lot of methane and it made the globe cool a lot. So climate change is not a new thing. Human climate change, which we think about, is a new thing, and just we are causing all this trouble, and I'm sure everyone was enjoying the heat wave last week. So human activity absolutely is causing disastrous impacts on the planet. But in the history of things, these impacts have happened due to microbial activity, and it's still happening today. So microbes affect the climate a lot. And I'm just going to talk about one thing called the DMSP cycle. So this is something that you may not have heard of. You may have heard of carbon dioxide as a climate warming gas, but DMSP is something that is really important in microbes and in the ecology of the Earth. So DMSP is a molecule that microbes produce. So plankton and things in the sea produce this. They use sunlight and sulfate, and they make this molecule called DMSP. And they use it for lots of things. It's a really useful thing that they use for protection from things, as food sources, all sorts of bits and pieces. And they make billions and billions of tons of this every year. But they also release some of it, and then other bacteria take that and they break it down. And one of the things they make is a gas called DMS. DMS gets into the atmosphere, and actually what it does is it nucleates clouds. So DMS promotes cloud formation and rain, and it reduces temperature. And this is important because as the sea level, or the temperature of the sea is changing due to human climate change, the activity of all of these processes is changing as well. So there's active work happening from my colleagues, John Todd, trying to understand what this means and what is this going to do for the climate as we go forward, because this microbial backdrop is really, really important. And if they change their activity and they're making more of the DMS or less of the DMS, that's going to have an enormous impact on climate change. And potentially we might also be able to make change for our benefit. Okay, so healthy planet. Bugs are important to the planet. Healthy plants. Plants are green because some point in history that cyanobacteria that evolved to do photosynthesis was stolen by plants. And so the chloroplasts inside plants is an example of what we call endosymbiosis, where a bacteria was taken up by the plant and now lives with it. So they both get benefit from it. So the photosynthesis in plants is done by a bacteria, essentially. Plants are important, as I'm sure we agree, for the food chain. No plants, no food, right? So plants are absolutely important. And one of the things that allow a lot of plants to grow is the fact that they have these endosymbiotic bacteria. So all plants have chloroplasts, which are bacteria allowing them to photosynthesize. But lots of plants have these things called root nodules. So lots of crops, the leguminous crops, so peas, beans, things like that, they all rely on bacteria in their roots. This is clover, and you can see these little lumps in the soil. These lumps are nodules that are packed full of a bacteria called rhizobia. And what this bacteria is doing is the plant is giving it sugar and giving it an environment to live. And what this bacteria is doing in here is it's pulling nitrogen out of the atmosphere. And it's doing something called fixation of nitrogen. Plants can't do that very easily themselves. They have to pull nitrogen up from the soil. You need nitrogen to make amino acids, to make proteins, to basically building blocks of your body. So plants are absolutely reliant, or some plants, on bacteria living with them to do this. And so this is a relationship that's evolved over billions of years to allow these plants to grow healthily. But not all interactions are good. So microbes do also cause enormous damage. So here is 3 examples of microbes that are causing damage to crops or trees. So on the left, this is rice, and this is rice that's been infected with something called rice blast. This is a fungus that causes enormous damage, and about a third of rice crops can be lost due to this. In the middle, we have another fungus growing on ash, and this is called something called ash dieback, and we're going to lose potentially all of the ash trees in the UK. They're all dying, okay? So this is something that emerged in Southeast Asia and has spread to Europe in the last 20 years, and between 90–95% of all of the ash trees are infected, and they're all going to die at the moment as things stand due to this fungus. This is potato, and this is something called potato blight, where there is a mold that can cause the potato to basically go off, and the actual fruit, if you like, is no good whatsoever. This caused the Irish potato famine, which you may have heard about, and caused enormous amounts of trouble to the rural communities who were absolutely reliant on potatoes as a food source. So these microbes can cause lots of trouble, but there is also some hope. So this is some work, again, that's been done at the Sainsbury lab in Norwich. And here we've got an example of the potato blight. So at the top, we have just a normal potato, and then one that's been infected with the mold that causes blight. You can see that's what it does to the potato, and no one's going to want to eat that. Okay, so that's what happens to the crop. That's no good. At the bottom, we've got the same thing. So again, you've got an uninfected potato, but this one has been infected, but this one has been precision engineered. So what we've done, or the guys at the Sainsbury lab done, is they've found a gene that you can give to this potato that turns on its immune system and recognizes the pathogen. So plants have immune systems, but normally they don't recognize the pathogen that causes the blight. But in this case, you can add this single gene in, and now this crop is not going to be affected. Okay. So this is an example of how you can use genetic engineering or precision engineering to develop crops that are resilient to pathogens. And these pathogens are causing trouble in all sorts of crops, and they're always evolving. So we need to try and use these techniques to stay ahead and keep feeding the growing population. Okay, people. So we also care about people. And as we sit in this room, you are all definitely as microbial as you are human. So all of you will have at least one microbial cell for every cell of your own. More than 95% of the DNA you all have is microbial. Okay? So there are more bacteria, or the same number of bacteria in your mouths now than people who have ever lived, all of you. Okay? And that's kind of normal, fine. So this is what we call our microbiome. So your microbiome is something that, these bacteria live on us and in us, so the bacteria in your skin, in your mouths, in your gut. And most of the time, this is absolutely normal and fine. So this microbiome contributes to you. It helps you digest your food. It helps keep your kind of skin healthy. So these bugs are not causing any trouble. But things can go wrong. So the gut is unbelievably rich in microbes. Okay, so you have an incredible abundance of microbes living in your gut. It's not that surprising. It's warm, it's moist, you shove food in it all the time. It's a lovely environment for microbes to grow. So you have incredibly rich diversity of microbes. However, sometimes this can become what we call out of balance, and we can have problems where some bacteria, or it doesn't have to be a bacteria, but often it is a bacteria, will overgrow and it will start causing damage and disease. So one example of that is something called Clostridium difficile. So this is a bug that lives in lots of people's guts, but normally it's at a very, very low level. But if you have an antibiotic or something, then actually lots of your bacteria get knocked back. C. diff doesn't. It will then grow, and it produces a toxin that creates chaos, basically, and you end up having a really horrible disease, really nasty diarrhea, and it has quite a high death rate, particularly in the elderly. And it also causes outbreaks in hospitals. So C. diff, you may have heard about it in the press, has caused thousands and thousands of deaths in the UK and around, and lots of outbreaks. So it's a really nasty disease. But there is a solution. So one thing that you can do is that when you have this loss of the normal microbiota that should be there, you can restore it. So you can literally give someone what we call a fecal microbe transplant. So you can give someone a transplant of fecal material from somebody else who has a normal, stable gut microbiome, and that will give them reset and reduce the symptoms. And with people who have C. diff, it's a really dangerous disease and it recurs a lot. But 90% of people who are given this FMT, this fecal transplant, 90% of the people are cured. So it works really, really well. And what's happening at the moment, again, this is happening in my institute, the Quadram, is trying to turn this into what we call a crapsule. So instead of having, again, I didn't come up with the name, but it works, right? And you get the idea. So at the moment, what happens, or what's historically happened, is you have to have a tube put down or up. It neither is lovely to deliver the FMT. But actually, if you can take a capsule of the bacteria that you need, then this is obviously nicer for people. So this is what's going on. Now, what happens in our gut doesn't just stay in the gut. We also have a lot of work and a lot of interest now in knowing that what happens in our gut can affect our health in lots of other ways, okay? So there's a thing called the gut-brain axis, where we know that things that happen in your gut microbiome can affect your cognition, okay? So what happens in the gut has been linked with loads of other diseases, including various diseases of cognitive performance and lots of other things. So we're very interested in trying to understand how. So what happens in your gut will affect the metabolites that get into your bloodstream, and that can affect function in other parts of your body. And here's one example I'm going to show you from a colleague of mine called Amy Parker, and she's been interested in trying to see how what happens in your gut affects cognition, but also visual performance. Okay, so this is what she's been looking at. So what she's been doing here is looking at the health, essentially, of eyes of mice that have been given different interventions to their microbiome. So just very briefly, this obviously that's an eye, and what we're looking at here is just a little slice from the back of a retina, and the blue dots you can ignore, they're just staining cells. The green dye is binding to something, a part of something called complement, which is an immune response. Basically, inflammation means that something is going wrong and that the cells are angry and it's not good. So the more green, the worse. So in this top left, this is what looks like in a young mouse's eye. So there's very little green. So a young mouse's eye, there's not much inflammation. If you look at an old mouse, you can see there's loads of green. Okay, so this mouse is unhappy, the eye is inflamed. That inflammation will end up in a reduction in function. Now, if you take a young mouse and you give it a fecal transplant into its gut from an old mouse, then you can see, unfortunately for this young mouse, you then generate all this inflammation. Okay. So you haven't done anything to its eyes, but you're affecting what's happening in its gut, and these two things are linked. But really interesting and excitingly, if you do the reverse, if you take an old mouse, which we know normally has a disrupted lots of inflammation in its eye, and you give it a fecal transplant from a young mouse, you really reduce that, that inflammation. They also get less stupid, which is good. So when you kind of present them with kind of cognitive tasks. So there's a lot of interest in how changing your microbiome can affect your cognition and other parts of your health. So there's lots of research into that, which is really promising and really interesting for kind of trying to get everyone to live longer, more healthily. Okay, one last area I want to talk about is antibiotics. So, I mean, hands up who's had an antibiotic in their life? It's pretty much everybody, right? So that may have just cured something that was annoying. It may have saved your life, okay? And we rely on antibiotics for modern medicine. So we need to treat infection, but also you can't treat someone with cancer without an antibiotic if you're going to do anything that immunocompromises them. You can't do a heart transplant. You can't do surgery. You need antibiotics for everything, right? And we've only had them for kind of 100 years or less, really. And in the golden age of antibiotic therapy, this is an old advert from Glaxo who are kind of trumpeting that we've got this range of antibiotics we're discovering. They're all being available, and they can treat all these things. That's great. Antibiotics have revolutionized medicine and put years and years and years on average life expectancy. However, you may well have seen headlines like this: superbugs threaten return to dark ages. Okay, so unfortunately bacteria evolve. This is my particular research interest. And bacteria are rapidly evolving resistance to antibiotics, and there are bacteria now who are resistant to all of the antibiotics that we have. And in some parts of the world, that's very common. It's not the most common in the UK, but we see examples of this. Our local hospital has had some examples where, yeah, what do we do? None of our antibiotics work against this bug. What are we going to do? So this is a real problem, because we're going to go back to where we were 100 years ago, where a kind of scratch or something might be life-threatening, where you would kind of consider if it got infected now and it got bad, okay, we'll just get some antibiotics and knock it on the head. So this is a big worry, a big problem. But again, there is some hope. I'm trying to balance all these stories with kind of hope at the end. So antibiotics have not been invented. Okay, we discovered these. These have been made by microbes. So microbes make antibiotics to kill other microbes because they are competing for each other. This is a bug called Streptomyces, which is an interesting bug. And our colleagues at the John Innes Centre work on this extensively. And about 70% of the antibiotics that we use in people were originally found in a Streptomyces. So this is a group of, there's lots of different species of Streptomyces. They live in the soil. The soil is packed full of bugs, even more than the gut. They're fighting each other. Okay, so we've discovered a whole bunch of antibiotics that these guys make to kill each other and then turn them into medicines we use in people. What we're doing now, though, is now we're able to sequence genomes and isolate organisms much more easily. We can now look much more broadly into these organisms, and we can see that they have hundreds and hundreds of gene clusters that probably are making antibiotics that we don't know what they are. So what we're trying to do now is unlock this to say, actually, can we take these and can we make the bug make them and find out what they do? So can we find hundreds of new antibiotics to get over this problem of resistance and allow us to start being able to treat things as we go forward and not return to the dark ages? Okay, so that is pretty much where we're going to end. I just wanted to kind of give you an overview of this and hopefully convince people that studying microbes is important or essential to understand how to protect healthy planet, healthy plants, and healthy people. And just a little plug, if you haven't been already, up at the top floor, there is a microbe zoo exhibit where you can go through interactively different environments that represent the gut, the soil, and kind of things that are happening in plants, where you can explore some of these concepts in a bit more detail. There's a whole bunch of guys there that can take you through it. But that is all I wanted to say. Hopefully that's about right for time. There wasn't any angry waving, so I think I'm just about okay. I'm very happy to have any questions, Atari. Thank you very much.
So I was just wondering, are you looking at bacteriophages at all instead of antibiotics?
Yep. So antibiotics are one way to kill bacteria. They're not the only way. Phage are, as I said, about a third of the bacteria in the sea get killed every day by phage. So in one way you think, wow, what a potent weapon, we could kill all the bacteria. The flip side of that is they bounce back the next day. So that, So phage in some so these are the viruses that kill the bacteria. They've got quite a long clinical history of being effective for certain things, and they work pretty well, and there are people actively progressing these. For things where you can get easy access to the infection, so if you've got wound infections or lung infections, they're really promising and probably will become a big part of treatment. Some other things where you've got sepsis, where bacteria circulate in the blood, delivery is a bit difficult. So they're probably not a panacea for everything, but absolutely they will be. I would expect to see phage therapies in the NHS in the next 10 years for various things, I would think. We use them in food production a lot already, actually. So about a third of the potatoes you eat have been sprayed with a bacteriophage to stop them going off so quickly.
We hear that development of antibiotics is commercially uninteresting. Is there any serious work going on?
Yeah, so the history of that is that antibiotics were developed quite a long time ago, most of them. They're also the only group of pharmaceuticals that you only use for a very short period, generally. So you might use them, you expect to use them for maybe a week or two. You expect them to be really cheap. And also, they're the only pharmaceuticals that the target will become resistant to and they'll stop working. So if you're a shareholder in a pharmaceutical company, disastrous, right? Why would you spend your money doing that? Develop a statin, something that someone takes a pill every day for 50 years, right? So that was the kind of problem. But there's been a lot of initiatives to try and unblock that. A lot of kind of public-private initiatives, but also changing the subscription model so you don't have to rely on kind of selling per pill. And there's an idea of a kind of Netflix model for drugs where, you know, kind of the NHS or other providers will say, yeah, we will subscribe to a whole bunch of drugs and we'll have, you know, a statin, a beta blocker, whatever. But in that, we must have an antibiotic as part of that package. So the incentive then to say, if you want to get paid for the package of drugs, you've got to have an antibiotic in there. Therefore, there's a commercial value to developing it. So these kind of pull initiatives are being developed to try and do it. There are companies trying to develop antibiotics, but when I first started doing this, in my PhD, was actually sponsored by Bayer, who gave up on antibiotics. Most of the big pharma companies gave up in the 90s, but not all. So it's, yeah, shoots of optimism, but also, yeah, we're not going to be flooded with 50 new drugs in the next 10 years.
What should hospitals and individuals be doing to slow down antibiotic resistance?
So hospitals and individuals and countries nationally, what you should do is use antibiotics kind of judiciously. So where there are policies in place, you should try and follow them because they've been set up to say, if you don't need to use an antibiotic, you shouldn't use it. So if someone's just feeling a bit unwell and you don't know if it's viral or bacterial, do you need to use it? If you don't, then you shouldn't. And you can see really clearly parts of the world where antibiotic use is controlled have much, much lower resistance rates than parts of the world where it isn't, right? So it definitely works. Obviously, when you do need to use a drug, you do need to use the drug. So individuals, you should use it. And if you're prescribed a course, finish the course as it's meant to, because quite often you'll take it for a day or two, you'll start feeling better, and then be like, okay, I'm going to forget taking those pills for the next 3 days. But actually what's happened is the number of bacteria has dropped down, but they haven't completely gone. And if you stop taking them, they all bounce back up again. And the more times you do that, the more chance there is for a mutation to happen that makes it resistant. So you're breeding, you're literally conducting an evolution experiment in yourself. So basically, just try and use drugs when prescribed, as prescribed. And in a hospital, they have policies where they should know what their local resistance rates are and which drugs are most likely to be effective and not. So yeah, it's tricky, but yeah. You can kind of minimize resistance reasonably if you follow good practice.